Let's start with what these sources present as a foundational concern: the engineered spike protein itself.
Yeah, that seems to be the starting point in most of these papers. They begin by explaining the basic mechanism.
Which is?
The mRNA vaccines instruct your cells to produce the SARS-CoV-2 spike protein. That's the core idea.
Okay. And a key claim raised in this material is that the engineered spike protein produced by the vaccine isn't quite the same as the spike protein on the natural virus.
That's a central point they make, yes.
So what differences do these sources highlight?
Well, they detail several specific properties of the engineered version. One is that it's described as being stabilized in an open conformation.
Open conformation. What does that mean in simple terms?
It means basically it doesn't fold back into the cell membrane in the way the natural one might after binding. It sort of stays exposed.
Okay, so it's presented as staying in a more active state, potentially.
That's the implication discussed, yeah. These sources also suggest it binds particularly tightly to the ACE2 receptor.
And ACE2 receptors are everywhere, right? Lungs, heart, blood vessels.
Pretty much. And these sources claim that by binding so tightly, it essentially disables or interferes with this receptor's normal function.
Which could lead to problems.
They link this within the sources to potential dysfunction in vascular, cardiac, and neurological systems. That's the connection they draw.
And they also raise concerns about the amount and duration of protein production. Is that right?
Yes, absolutely. The material points to the high GC content. That's a technical term for the RNA sequence in the synthetic RNA used in the vaccines.
And what's the significance of that, according to them?
According to these sources, this leads to excessive and, importantly, prolonged production of the spike protein by the body's cells.
How long are we talking?
Potentially for weeks or even months, which they claim far exceeds the more transient production you'd see during a typical natural infection.
So the concern highlighted in these sources isn't just making the spike protein, but making a potentially modified version.
Right.
Possibly in much larger quantities and for much longer periods based on the claims in these documents.
Precisely. And these unique properties, as they describe them, are presented as contributing to significant downstream effects.
Such as?
The sources mention inflammation and mitochondrial damage as potential consequences.